Last year. Okay, well. So Ahmed's been on board a year. So what have you done? So anyway, Ahmed got his undergraduate degree in Egypt, his master's degree in Egypt, and then he went to the University of Manchester, right? And he worked with a famous person there on Keck, right? Who was an expert on placental morphogenesis and development, and actually is now switched into stem cell biology. Anyway, he did a great job in PhD. He then went to New York and didn't like it very much. and then he came to work with me, and he did extremely well on postdoctoral studies and got 4 or 5 really high impact papers, and he's opened up a whole new field, which is the biology and behavior of lung progenitor cells in the embryonic lung, which sets the whole thing going. Because if that doesn't work right, you're screwed, right? So Ahmed, please tell us all about your latest stuff.
Oh, thank you so much. Thank you so much.
Speak up, because okay.
So good afternoon, everybody. My talk is about the molecular control of stem or progenitor cell behavior during lung morphogenesis. Yeah, take some background about lung development. Lung development starts as 2 primary buds which undergo stereotypic round of growth. So lung development starts as 2 primary.
You must have pressed the arrows when you press the red buttons.
So lung development.
Sorry. Go ahead. I tried it all.
Right.
So lung development starts as 2 primary buds which arise from the Regenerative group and the ventral flow of the primitive frontal. They undergo stereotypic round of branching to form this complex organ which is the lung. The epithelial stem cells are localized at the distal epithelial tip during embryogenesis and can be easily identified by the expression of some genetic marker like ID2, which is specific for them, and also SOX9 and NMET. The mechanism which controls the behavior of this stem is still unknown. One of the key factors which has been discovered to control the behavior of stem cells is called EY1 protein phosphatase. EY1 stands for I-absent, which is belongs to protein tyrosine phosphatase BTBT family. It contains phosphatase activity, which is essential for the regulation of progenitor cell proliferation and also for directing cell to repair illicit apoptosis after DNA damage, and also for different EYA cytoplasmic serum function. Little is known about EY1 target, and the function of EY1 in the lung is still unknown before our studies. In the absence of EY1, there are POR syndrome, which is characterized by defect in inner ear development, and therefore there is hearing loss, there is no kidney, and there is, and also there is a lung defect, which I'm going to talk about. Here is the expression pattern of EY1. It is expression of EY1 starts early during development from day 10, and it is highly expressed until days 14.5 and 15.5. And this phase is characterized by a lot of stem cells. There is a lot of branching in this phase, which again suggests that EY1 might be important for the branching morphogenesis. After that, start from day 17 and 18.5, the expression is down-regulated, comparable to the expression of differentiation marker, which again suggests that EY1 inhibits the differentiation of this stem cell. EY1 is specifically expressed as the distal epithelial tip, which contains the stem cell. Therefore, you can consider it as a marker for this stem cell. It has also expression in the distal mesenchymal cell, which again is another kind of stem cell in the middle card. But I'm going to talk about its function in the epithelium. This distal epithelial tip expresses the SOX9, which is a marker for progenitor cell, and And the expression of EY1 is unique because it has a polarized localization at one side of the cell, which again suggests that EY1 might be important for the process of epicanthesal polarity. In the absence of EY1, there are severe lung hypoplasia, which is characterized by small size lung. You can see at 14.5 and 18.5. There is a reduction in the number of branching. There is only 5 branches in the mutant at 14.5 compared to 17 branches at the same stage in the control. There is also arrested expansion of epithelial tube in this mutant comparable to the control wild type, and there is a dense mesenchymal cellularity. There are 2 key features of EY1 phenotype. The first one is there is a severe reduction of epithelial stem cell, expressing SOX9 and IB2. At the same time, there is a significant increase in the differentiated cells at 18.5, and this differentiated cell express SCBC, SCBP, and other differentiation markers. And this data suggests that there is a defect in the cell proliferation of this epithelial stem cell. To study this point further, I carried out mitotic index analysis by staining for phosphorescence 3. And as you can see here in the 3 mutant of EY1, there is a significant reduction in the mitosis of this epithelial stem cell. We know that from cell cycle study that when cells cease division, they undergo either differentiation or apoptosis. So to study this point, what happened to the cell cycle in this mutant, I carried out cell cycle exit analysis. In this experiment, I inject the mice with BRDU, and after 24 hours of BRDU injection, cell cycle exit can be estimated as the ratio between BRDU positive, PI6 stable negative cells, and the BRDU positive cells. You can see here in 3 different mutants, there is a significant increase in the number of cells that exit the cell cycle. At the same time, there is no significant increase in apoptosis, but indeed there is a significant increase in differentiation. And this data suggests that there is a defect in the balance between cell renewal and differentiation of epithelial stem cell in the absence of EY1. So what's the importance of this balance? The balance between stem cell renewal and differentiation maintain tissue homeostasis. Abnormal increase of stem cell renewal may lead to the tissue hyperplasia or also tumor formation, while the excess of differentiation may lead to tissue aging or tissue degeneration. The mechanism, or the basic mechanism, which maintains this balance between cell renewal and differentiation is asymmetric cell division. Because asymmetric cell division produces one daughter cell which acquires stem cell fate and another daughter cell which has a differentiation fate. There are 2 types of cell division, symmetric and asymmetric cell division, based on the behavior and segregation behavior of the cell fate determinant protein, like which, for example, NAM, which I'm going to talk about it in the next few slides. If this cell fate determinant, which one is in blue, is inherited by both daughter cells, they will acquire the same fate. And this is an example of symmetric division. While in the case of asymmetric cell division, the same fate determinant is inherited by only one daughter cell. In case of NAM, this daughter cell will acquire differentiation fate, while the other one without NAM will stay as a polygonal or stem cell. There are 2 mechanisms of asymmetric cell division. Depending on whether this cell fate determinant is intrinsic or extrinsic. In case of extrinsic fate determinant or microenvironment, the 2 daughter cells are placed in 2 different microenvironments and therefore will acquire 2 different fate. While in case of intrinsic fate determinant, which I'm interested in, the cytoplasmic fate determinant protein like NAM, the one in green, has one in blue, has a polarized localization in the dividing cell. And when this cell gives rise to daughter cell, NAM is inherited by one daughter only. And therefore, this daughter will acquire different fate from the other. And here is some information about NAMP. NAMP contains amine terminal P2B domain and carboxy terminal endocyclic binding motif. There are 4 mammalian NAMP splicing. NAMP acts as a negative regulator of notch signaling by promoting the degradation of notch. And here is a model of NAMP action on notch and on cell fate. Normally, in wild-type stem cell, during division, NAMP has a polarized localization, which is critical for NAMP function. Loss of priority of NAMP affect its function and will therefore affect on whether the division is symmetric or asymmetric. And I will show you some data about it. So normally NAMP has a polarized localization. When cell divide, NAMP is inhibited by one daughter cell only, and this daughter cell NAMP acts to inhibit notch. And therefore, in different systems, we know that notch acts to maintain the identity of stem cell. So by inhibiting